Welcome back for more AP Biology. In particular, we're going to deal with active transport and its results, which are membrane potentials. So we're going to try and deal with, well, what's the ramification of all of this? And a whole bunch of stuff. Sorry, just did the lecture in class and I did some shouting. So one of the things that happens with a membrane is its job is to regulate what's allowed in and out of the cell. If you have something that's nonpolar, like oxygen gas, you don't get much say in that. But other molecules that are smaller than proteins, you actually get some control. You get some control over that. And that's going to be through membrane proteins. The catch is, how do we make active transport really happen? And what we have to do is provide energy to that in the form of ATP. ATP turns out to have 3 phosphates all stuck in a row. And each time you add a phosphate on, it's a little more difficult to actually make that attachment. So 1 phosphate, it's okay. 2 phosphates, kind of difficult. 3 phosphates, this is really difficult. So what happens is a lot of energy is put into attaching that last phosphate. The result is that connection, that bond, is very, very, very energetic. So if I were to let it snap, I let energy out. If I just did that willy-nilly, I get an explosion. Cells don't like explosions, so what you do is you direct the energy of the breaking into forming other things, to making other reactions occur. In class, what I did was I then walked through, well, here's really how that would work. But first, proteins are also picky. Meaning, if I were to take, kind of like when you were siblings and you had fingers and you were getting close to your sibling in the car and then eventually, he's going to touch me, he's going to touch me. Well, that is just getting near other proteins causes the protein to react. It changes its shape. So if you actually were to touch a protein, you'll get probably a very big change in what it looks like. Likewise, if I were to remove that protein, remove whatever the attachment is from the protein, I will also get a really big change in its shape. So what I can do is, if I need to have a lot of shifting, that's going to take some energy. So what if I could use ATP and breaking that last phosphate off, using that energy to make the protein change its shape? And that's exactly what happens in active transport. Obviously, this is a blank screen. In class, what we did was I actually showed where we actually drew out, here's active transport. And what I'm going to do is I'll take a picture of basically what we did and I'll put it into here so you actually don't see a blank screen. You'll actually see what we resulted in. But what it turns out that we can do is if we use a little bit of trickery to get sodium ions and potassium ions to bind to parts of a protein. This is for something that we would call the sodium-potassium pump. And we use ATP and the energy found inside of it to make sodium ions be moved out of the cell and to drag potassium ions into the cell. But if you weren't here for class, ask your friends for how that drawing was ultimately done. All you can see right now is here's the drawing that we did. Why do we do this? We have 3 results. Result number 1 is we can stockpile. We can hide certain elements into certain parts of cells, which means we can concentrate up ions for some purpose. Second reason requires some background. First one is all things come in pairs in terms of charge, so every positive charge has a negative charge. And we know that positive and negative charges attract each other. That's referred to as Coulomb's law. Turns out by moving sodium ions and potassium ions, I'm setting up concentration differences or concentration gradients. Through mechanisms we don't care about right now, there is an unequal distribution of plus charges and minus charges across that membrane. Almost universally, because I'm sure there's an exception to this, I just don't know it, the outside of the cell has a positive charge and the inside of the cell has a negative charge. So what we get is what we would call an electrochemical gradient. The outside of the cell has sodium. Sodium wants to move into the cell for 2 reasons. Reason number 1, concentration-wise, you move from high concentration to low concentration according to diffusion. Second reason is the inside of the cell is negative. So the positively charged sodium ions want to move to the negatively charged inside of the cell. That phenomenon, that electrochemical gradient, is also sometimes referred to as a membrane potential. Membrane potentials are a nice way of saying a battery. your cells turn out to be batteries. And we actually can do all sorts of things using this particular fact that we're a battery. Among them, with this one here, which is relatively simple, is we can use the membrane potential to make cells move, like with Bacterium and Flagellum, or with bacteria and us, we can use it for respiration. In cell respiration, turns out we can end up generating membrane potentials. In photosynthesis, with other eukaryotes, not us, we can actually use photosynthesis to make a membrane potential, and both of those turn out to be used to make ATP. We also use membrane potentials to make our brains work. We use membrane potentials to make our muscles work. We use membrane potentials to make plants open and close openings that they happen to have on their leaves that we call stomata. We actually use these membrane potentials for all sorts of things. So this is ramification number 2. First ramification is we can stockpile. Second ramification is we have membrane potentials, which can be used for things. The third one is what we would call symporters or secondary active transport. So what I can do is use active transport to move sodium ions, like moving sodium ions outside of the cell, as we can see with several of these proteins. But if I want to let that sodium ion back into the cell, because electrochemical gradient says move back into the cell, I can actually coerce it to bringing another molecule with it that I want to force into the cell. So like the first example, chloride ions don't naturally go inside of the cell. So what you do is you say, I'll let the sodium in, but only if the sodium brings the chloride with it. And the result is sodium and chloride both enter the cell. We also do the same thing with like glucose. I use sodium to bring glucose into the cell.